We've just touched upon the potential for collective power in neural systems. Now, let's zoom right into the microscopic messengers that make this collective action possible. Neurotransmitters. You know, those tiny chemical couriers shuttling signals across the synapse. But here's the kicker. It's not just one type of courier. Why complicate things? Well, imagine trying to have a nuanced conversation with only two words, yes and no. You'd quickly run out of descriptive power. Similarly, having multiple neurotransmitters allows for a richer, more complex dialogue between neurons. Now, these neurotransmitters aren't all created equal in their effect. Some are like the party starters. They push the neuron towards firing, making its inside less negative. We call these excitatory neurotransmitters. Think glutamate, the main excitatory player in your brain. Then you have the chill pills of the neurotransmitter world, inhibitory neurotransmitters. These guys do the opposite. They make the neuron even more negative, pushing it further away from firing. GABA is the big name here, the primary inhibitory neurotransmitter. So, a single neuron isn't just getting simple yes or no signals, it's receiving a constant barrage of different volumes and even directions, some pushing it towards action, others pulling it back. It's like being at a crowded bar trying to listen to one conversation while ignoring all the noise around you. That integration process at the axon hillock, it's where the magic or the madness happens. Okay, so if every neuron potentially releases a whole cocktail of neurotransmitters, how do we keep track of who talks to whom? Enter Henry Dale, a true giant of the field, and his eponymous principle. Dale basically said, hold on, let's keep it simple. His rule, proposed way back in the 30s, states that when a neuron fires, it releases only one specific type of neurotransmitter from all its connections. No mixing and matching per action potential. This was revolutionary because it meant each neuron had a signature, a unique neurochemical identity. If neuron X is a serotonin neuron, then all the neurons it connects to must have serotonin receptors waiting for it. It imposes a kind of order on the apparent chaos, ensuring that communication follows predictable pathways based on these distinct chemical identities. It's the bedrock assumption that makes mapping neural circuits feasible. So, how does the brain manage to produce these essential chemical messengers efficiently? It turns out there are some pretty clever design principles at play, driven by necessity. First, availability. Imagine you're running for your life from a lion, and your brain runs out of neurotransmitter for your leg muscles because it couldn't synthesize it fast enough. Not ideal. So, the body uses precursors that are readily available, often straight from your diet. Need serotonin? Grab tryptophan from your turkey sandwich. Need dopamine? Tyrosine is your friend. It's like having raw materials on hand for urgent construction projects. Second, speed and efficiency. Neurons can fire dozens of times a second. You can't afford to build massive, intricate neurotransmitter molecules every single time. The synthesis process needs to be quick, like snapping together Lego bricks, not carving marble statues. 2 steps to turn tyrosine into dopamine? That's efficient. Finally, economy of scale. Why reinvent the wheel? If you can make dopamine, why not also easily convert it into norepinephrine using another enzyme? It's all about minimizing waste and maximizing output for these fleeting chemical signals. Cheap, easy, and fast. That's the neurotransmitter playbook. Now that we understand the basics of neurotransmission, we can start looking at how drugs interact with this system. Think of neuropharmacology as essentially using chemicals to tweak the brain's communication lines. It's a double-edged sword, capable of both healing and harm. Broadly speaking, these drugs fall into 2 camps. Those that amplify the signal, essentially turning up the volume or increasing the number of messengers, and those that dampen the signal, turning down the volume or blocking the message. Understanding these mechanisms is crucial, whether we're talking about treating depression, managing pain, or understanding the effects of recreational substances. It's all about manipulating the delicate balance of excitation and inhibition. So, how do we crank up the signal? There are several strategies. One is to boost production, either by giving the neuron the raw ingredients, the precursors, like L-DOPA for dopamine in Parkinson's disease, or by using drugs that supercharge the enzymes responsible for synthesis. Another approach is direct intervention. Give the neurotransmitter itself, or a synthetic lookalike that can fool the receptor, like psilocybin mimicking serotonin. Increasing the number of receptors on the receiving neuron would also work, but unfortunately, that's notoriously difficult to achieve pharmacologically. A very common and effective method is to stop the cleanup crew. Many neurotransmitters are broken down by specific enzymes or reabsorbed by transporters right after they do their job. By blocking these degradation or reuptake processes, you let the neurotransmitter linger longer in the synapse, hitting more receptors. This is the core mechanism behind many antidepressants like Prozac, which are selective serotonin reuptake inhibitors or SSRIs. They selectively block the reuptake pump for serotonin, leaving more serotonin floating around to do its work. On the flip side, how do we turn down the signal? The logic is similar, just reversed. You can block the production line entirely, preventing the neurotransmitter from ever being made. Or, you can jam the release mechanism, stopping the vesicles packed with neurotransmitter from fusing with the membrane and spitting out their cargo. A very common strategy is to block access to the receptor. This can happen in different ways, like using a competitive antagonist, a molecule that looks like the neurotransmitter but doesn't activate the receptor, essentially sitting in the receptor's seat and blocking the real neurotransmitter from getting through. A classic, albeit deadly, example is Curare. Used historically in hunting darts, Curare blocks acetylcholine receptors at the neuromuscular junction. Acetylcholine normally tells your diaphragm muscle to contract for breathing. Block those receptors, and you can't breathe. It's a stark reminder of how critical these signaling pathways are for basic survival functions. Now, here's a crucial point that ties everything together. The brain isn't a fixed, unchangeable machine. Experience actively reshapes the very machinery of neurotransmission. Just like the threshold for firing an action potential can change, almost every aspect of neurotransmitter function is plastic. The number of receptors on a cell can go up or down, a process called upregulation or downregulation. Think of it like turning the volume knob up or down. The sensitivity of those receptors can change too, how tightly they bind the neurotransmitter. Even the rate of synthesis, release, breakdown, or reuptake can be adjusted based on recent activity. It's a dynamic system constantly tuning itself. This plasticity is fundamental to learning, memory, and adaptation. It means the brain isn't just reacting to the world. It's actively modifying its own communication channels based on what's happening. Let's move beyond simple pairwise interactions and consider what happens when three neurons get involved. This introduces the concept of neuromodulation. Imagine neuron A influencing neuron B, which in turn affects neuron C's standard stuff. But what if neuron A also sends a signal directly to neuron B's axon terminal, the very place where neuron B releases its neurotransmitter onto neuron C? That's what we see in this axoaxonic synapse diagram. Neuron A releases GABA onto neuron B's axon terminal. This inhibits neuron B right before it's about to release its excitatory neurotransmitter onto neuron C. So neuron A isn't directly talking to neuron C, but it's modulating the conversation between B and C. It's like whispering in someone's ear to tell them not to say something important. This allows for fine-tuning of neural circuits, adding another layer of complexity beyond simple excitation or inhibition. How does the brain create signals that are sharp in time? Think about it. A neuron firing continuously is hard to distinguish from a neuron firing intermittently. The brain loves contrast, being energetically loud when it has something to say and energetically silent otherwise. One way to achieve this temporal sharpening is through negative feedback loops. Look at this circuit. Neuron I excites both neuron B and neuron C. Crucially, neuron C then sends an inhibitory signal back to neuron A. So when neuron A fires, it activates both B and C. But activating C triggers a signal that silences A. It's like shouting fire, and then immediately putting a gag on yourself, so you can't shout again. This creates a burst of activity followed by a period of silence, making the original signal much clearer over time. Neuron A can even control how strongly it shuts itself up by deciding how many of its outgoing connections go to B versus C. Clever, huh? There's another way to sharpen a signal over time, using feed-forward inhibition. This is like predicting the noise and silencing it before it even arrives. Again, neuron A excites both neuron B and neuron C. But this time, neuron C sends an inhibitory signal to neuron B. Now notice the timing. The path from A to B is direct and fast. The path from A to C to B involves two synapses, so it's slower. What happens? When neuron fires, neuron B gets excited immediately. Neuron C also gets excited, but it takes a bit longer for C to process that and send its inhibitory signal to B. By the time that inhibition arrives at B, neuron B has already fired its response to the initial excitation from A. The result? The inhibition arrives just after the main signal, effectively silencing any subsequent chatter from B. It cleans up the signal, making the initial response sharper and more transient. It's like knowing there's going to be background chatter at a meeting and muting yourself beforehand to ensure your point gets heard clearly. Beyond time, the brain also sharpens signals in space. Think about looking at a dimly lit room. You don't perceive the light as uniformly spread out. You perceive objects and edges. How? Lateral inhibition. Look at this circuit involving neuron C. When neuron C receives an excitatory input, it does two things. It excites neuron D, and it sends inhibitory signals to its neighbors, neurons A and E. So stimulating C makes D fire, but it also silences A and E. The net effect? The signal from C stands out much more clearly against the quieter background of A and E. It's like shining a spotlight on one area while dimming the surrounding lights. This mechanism is incredibly important in sensory systems. It helps you pinpoint exactly where you were touched, enhances the contrast in what you see, and even helps distinguish musical notes. It's all about creating sharp boundaries and highlighting features within the sensory landscape. This elegant circuit model beautifully explains why we experience different types of pain. Think about the difference between a sharp needle prick and a dull, throbbing burn. This circuit proposes a mechanism based on the two types of pain fibers we discussed earlier: fast myelinated fibers like neuron A and slow unmyelinated fibers like neuron D. Both types of pain signals ultimately reach the spinal cord neuron B. However, both also activate neuron C, which plays a key role. Neuron A, the fast fiber, excites B but also activates C, which then inhibits B. This leads to a brief, sharp pain signal. Neuron D, the slow fiber, also excites B, but it activates C in a way that inhibits C's ability to inhibit B. It's a double negative. This allows the signal from D to pass through largely unhindered, resulting in a longer-lasting, throbbing pain. Interestingly, the slow conduction speed of D adds to this perception. This model even explains why scratching an itch can temporarily relieve a deeper ache. Scratching activates the fast pain pathway, briefly silencing the chronic pain circuit via C. Clinically, this understanding led to treatments like implanting electrodes to stimulate the fast pain pathway and shut off chronic pain. It's a testament to how circuit-level interactions shape our subjective experiences. Let's consider a hypothetical circuit that illustrates how the brain might learn abstract concepts. Imagine layer 1 neurons are specialized detectors, recognizing specific details, like the face of Elvis or the posture of a Victorian gentleman. Layer 2 neuron 3 receives inputs from multiple layer 1 neurons. Through convergent connections, neuron 3 learns the general concept represented by the overlap of these specific features. It becomes a generalist neuron, understanding the essence of Victorian gentleman dressing, even if it hasn't seen every single example. This kind of hierarchical processing, where higher-level neurons integrate information from lower-level specialists, is thought to underlie our ability to associate disparate ideas, form metaphors, and create symbols. It's the neural basis for linking the color of wine to the sea, or associating a pattern of colors with a nation. And perhaps, just perhaps, the breadth and idiosyncrasy of these associative networks in some individuals might be linked to creativity. Someone seeing faceness not just in a simple drawing, but in the fragmented geometric style of Picasso. Maybe that's their unique way of connecting the dots, drawing broader, more unconventional associations. We've talked about individual neurons, pairs, and small circuits. Now, let's scale up dramatically to the level of brain regions. Unlike the liver, which looks pretty much the same everywhere you slice it, the brain is incredibly organized. Neurons with related functions tend to cluster together in specific areas, forming nuclei or larger regions. And the axons projecting from these clusters often bundle together into massive fiber tracts, like cables carrying information between different parts of the brain. This internal organization is what gives the brain its functional specialization. Different regions like the hypothalamus, cerebellum, or hippocampus have distinct roles and names. This structure is key to understanding how complex behaviors emerge from the coordinated activity of millions, even billions, of neurons working together in specific locations. Those bundled axon cables we just mentioned? They form the white matter of the brain. Remember myelin, the fatty sheath wrapped around axons that acts like insulation, speeding up signal transmission? Well, myelin is pale white. So, when you look at a cross-section of the brain, especially deeper inside, you see these vast networks of bundled myelinated axons appearing white. This white matter constitutes a huge portion of the brain's volume. It's the infrastructure, the highways and byways, enabling communication between the various gray matter regions which contain the cell bodies. Without this extensive white matter network, the brain wouldn't be able to coordinate activity across its vast expanse efficiently. It's the connective tissue that holds the whole complex system together. Historically, one of the most powerful ways neuroscience advanced was through lesion studies. Think about soldiers wounded in battle. Tragically, their injuries provided invaluable insights. By observing what functions were lost due to damage in a specific spot X, researchers could infer what that region did. However, interpreting these lesions requires care. If spot X is a nucleus, a cluster of cell bodies, then the damage likely disrupted the function originating from those cells. But if spot X is a fiber tract, a bundle of axons passing through, the damage disrupts communication along that pathway, affecting functions related to both the source and destination regions. This distinction between neuronal nuclei and fibers of passage is crucial. And importantly, as we've emphasized throughout, modern neuroscience views brain function through the lens of networks. It's rare for a single region to be the sole center for a behavior. Instead, functions are distributed and embedded within interconnected networks. A region typically plays a key role in or influences a behavior rather than being solely responsible for it. Alright, let's shift gears slightly and talk about hormones. While neurotransmitters are the local couriers, hormones are the long-distance dispatchers. Released by glands into the bloodstream, hormones travel throughout the entire body, potentially reaching any cell equipped with the right receptor. This is a massive difference in scope compared to neurotransmitters acting across a tiny synapse. Furthermore, hormones operate on a different timescale. Their effects unfold slowly, over hours, days, or even weeks, unlike the near-instantaneous nature of neurotransmission. And the scale of their impact is often much larger. Hormones can fundamentally alter gene expression, trigger growth, cause cell death, or regulate metabolism system-wide. Think testosterone building muscle, thyroid hormone orchestrating metamorphosis, or stress hormones suppressing immunity. They are the body's systemic regulators, working in concert with the more localized neurotransmitter system. Most hormones don't work in isolation. They're part of carefully regulated neuroendocrine axes, often orchestrated by the hypothalamus. Think of the hypothalamus as the master control center, integrating information from the brain and body. A classic example is the stress response axis. Under stress, hypothalamic neurons release corticotropin-releasing hormone. CRH travels to the pituitary gland, stimulating it to release adrenocorticotropic hormone. ACTH then enters the general circulation and travels to the adrenal glands, prompting them to release glucocorticoids like cortisol. This cascade amplifies the signal from the brain to the periphery. It's a beautiful example of hierarchical control. And it's often even more complex, with multiple hormones potentially influencing the same pituitary hormone release, allowing for fine-tuning based on the type and intensity of the stressor. While the hypothalamus-pituitary axis is common, not all hormones follow this exact pattern. Some are regulated directly by the brain acting on the pituitary, like growth hormone. Others involve direct neural projections from the brain to the target gland, like the regulation of insulin release by the pancreas in response to blood sugar levels. There are even hormones secreted from unexpected places, like the gut or heart. Despite these variations, there are similarities to neurotransmitters. Hormones are also made cheaply from readily available precursors, like cholesterol for steroid hormones. And just like neurotransmitter receptors, hormone receptors are highly specific, fitting only their designated hormone like a key in a lock. Crucially, the number of receptors and their sensitivity can change, altering the cell's response to the hormone. This receptor dynamics is vital for adaptation and can even lead to diseases if receptors malfunction, even when hormone levels are normal. Hormones play a dual role in shaping us depending on when they act. During critical periods of development like fetal life or puberty, hormones have organizational effects. They literally shape the developing brain and body, setting up the infrastructure for future responses. These changes are often permanent. Think of hormones organizing the sexual differentiation of the brain during development. Later in adulthood, hormones exert activational effects. These are temporary influences, often related to daily physiological rhythms or responses to environmental challenges, like stress or reproductive cycles. These effects are reversible. The interplay between these two is fascinating. The organizational effects laid down early in life determine how the brain will respond to activational hormone signals throughout adulthood. It's a lifelong dance between prenatal programming and adult modulation. Let's take a quick detour into the molecular world of proteins. These are the true workhorses of the cell. From the hormones and neurotransmitters we've discussed, to the enzymes that build and break them down, to the scaffolding that holds the cell together, and the receptors that detect signals, proteins are everywhere. What makes a protein unique is its shape. Its three-dimensional structure dictates its function. An enzyme needs a specific shape to perfectly fit its substrate and catalyze a reaction. A receptor needs a complementary shape to bind its ligand. Many proteins are dynamic, changing shape upon binding, like a key changing shape as it turns in a lock. Ultimately, this incredible diversity of shapes arises from the sequence of amino acids strung together to form the protein chain. And that sequence? It's dictated by the genetic code stored in DNA. So, how does the information in DNA translate into the physical protein? It's a process often summarized by the central dogma, articulated by Francis Crick. DNA contains the code written in 4 nucleotide letters ATC. Groups of 3 letters called codons specify one of 20 amino acids. Because there are 64 possible codons but only 20 amino acids, the code is redundant, meaning multiple codons can code for the same amino acid. A gene is essentially a stretch of DNA that codes for one specific protein. To get the protein building instructions out of the nucleus where DNA resides and to the cytoplasm where proteins are made, the DNA code is transcribed into RNA. RNA is like a disposable copy of the gene's instruction manual. This RNA then travels to the ribosome, the protein factory, where it's translated into a sequence of amino acids, which then folds into the final protein structure. So the flow is generally, DNA determines RNA determines protein. Simple, yet profound. Life isn't always perfect replication. Sometimes mistakes happen when DNA is copied, especially during meiosis for gametes or mitosis for growth. These errors are called mutations. The simplest type is a point mutation, where just one nucleotide is swapped. Thanks to the redundancy in the genetic code, sometimes this swap doesn't change the amino acid produced, a silent mutation. Other times it changes the amino acid, but maybe only subtly, leading to a slightly altered protein. But sometimes that single change can be catastrophic, drastically altering the protein's shape and function. More disruptive are deletions and insertions. Losing or adding even a single nucleotide can cause a frameshift mutation, shifting the reading frame of the entire gene downstream, leading to a completely garbled protein sequence. Larger deletions or insertions can have similarly devastating effects, ranging from minor tweaks to complete loss of function or even death. These mutations aren't just errors, they are the raw material for evolution. When a mutation occurs, it creates a new version of a gene, called an allele. If this new allele confers a survival or reproductive advantage in a given environment, natural selection tends to favor individuals carrying it, leading to its increase in the population over generations. Conversely, disadvantageous mutations tend to decrease. Neutral mutations might persist at varying frequencies. This process generates the genetic variation we see within populations. For instance, we humans share about 98% of our gene types with chimpanzees, reflecting our shared ancestry and similar biological traits. However, when comparing any two human siblings, for example, we share about 50% of our gene alleles, the specific versions of genes. This 50% overlap reflects the individual variation arising from mutations accumulated over generations and passed down through inheritance. Finally, a quick look at the glossary of abbreviations used throughout the text. As you know, scientific writing, especially in fields like neuroscience and genetics, is rife with acronyms. From common brain regions like ACC or GABA, to techniques like fMRI or EEG, to disorders like ADHD or ASD, and genetic concepts like GWAS or BDNF, abbreviations are everywhere. This table provides a handy reference for many of the terms encountered. Knowing these helps navigate the literature and discussions in the field.